Class 11, Chapter 15, Body Fluids and Circulation. You have learned that all living cells have to be provided with nutrients, O2, and other essential substances. Also, the waste or harmful substances produced have to be removed continuously for healthy functioning of tissues. It is therefore essential to have efficient mechanism for the movement of these substances to the cells and from the cells. Different groups of animals have evolved different methods for this transport. Simple organisms like sponges and coelentrates circulate water from their surroundings through their body cavities to facilitate the cells to exchange these substances. More complex organisms use special fluids within their bodies to transport such materials. Blood is the most commonly used body fluid by most of the higher organisms, including humans, for this purpose. Another body fluid, lymph, also helps in the transport of certain substances. In this chapter, you will learn about the composition and properties of blood and lymph, that is tissue fluid, and the mechanism of circulation of blood is also explained herein. 15.1 Blood. Blood is a special connective tissue consisting of a fluid matrix, plasma, and formed elements. 15.1.1 Plasma. Plasma is a straw-colored viscous fluid constituting nearly 55% of the blood. 90 to 92% of plasma is water and proteins contribute 6 to 8% of it. Fibrinogen, globulins, and albumins are the major proteins. Fibrinogens are needed for clotting or coagulation of blood. Globulins primarily are involved in defense mechanism of the body, and the albumins help in osmotic balance. Plasma also contains small amounts of minerals like sodium, calcium, magnesium, HCO3- ion and Cl- ion, etc. Glucose, amino acids, lipids, etc. are also present in the plasma as they are always in transit in the body. Factors for coagulation or clotting of blood are also present in the plasma in an inactive form. Plasma without the clotting factor is called as serum. 15.1.2 Formed elements. Erythrocytes, leukocytes, and platelets are collectively called formed elements, figure 15.1, and they constitute nearly 45% of the blood. Erythrocytes or red blood cells RBC are the most abundant of all the cells in blood. A healthy adult man has on an average 5 millions to 5.5 millions of RBCs per millimeter cube of blood. RBCs are formed in the red bone marrow in the adults. RBCs are devoid of nucleus in most of the mammals and are biconcave in shape. They have a red colored iron containing complex protein called hemoglobin, hence the color and name of these cells. A healthy individual has 12 to 16 grams of hemoglobin in every 100 ml of blood. These molecules play a significant role in transport of respiratory gases. RBCs have an average lifespan of 120 days, after which they are destroyed in the spleen, and spleen is also known as graveyard of RBCs. Leukocytes are also known as white blood cells or WBCs, as they are colorless due to the lack of hemoglobin. They are nucleated and are relatively lesser in number, which average 6,000 to 8,000 per millimeter cube of blood. Leukocytes are generally short-lived. We have two main categories of WBCs, that is granulocytes and agranulocytes. Neutrophils, eosinophils, and basophils are different types of granulocytes, while lymphocytes and monocytes are the agranulocytes. Neutrophils are the most abundant cells, composing 60 to 65% of the total WBCs, and basophils are the least, composing 0.5 to 1% among them. Neutrophils and monocyte, composing 6 to 8%, are phagocytic cells which destroy foreign organisms entering the body. Basophils secrete histamine, serotonin, heparin, etc. and are involved in inflammatory reactions. Eosinophils 2 to 3% resist infection, and are also associated with allergic reactions. Lymphocytes 20 to 25% are of two major types, B and T forms. Both B and T lymphocytes are responsible for immune responses of the body. Platelets, also called thrombocytes, are cell fragments produced from megakaryocytes, special cells in the bone marrow. Blood normally contains 1,50,000 to 3,50,000 platelets per millimeter cube of blood. Platelets can release a variety of substances, almost of which are involved in the coagulation or clotting of blood. A reduction in their number can lead to clotting disorders, which will lead to excessive loss of blood from the body. 15.1.3 Blood groups. As you know, blood of human beings differ in certain aspects, though it appears to be similar. Various types of grouping of blood has been done. Two such groupings, the ABO and RH, are widely used all over the world. 15.1.3.1 ABO grouping. ABO grouping is based on the presence or absence of two surface antigens. Surface antigens are chemicals that can induce immune response, and these are present on the RBCs, namely A and B. Similarly, the plasma of different individuals contain two natural antibodies. Antibodies are proteins produced in response to antigens. The distribution of antigens and antibodies in the four groups of blood A, B, AB, and O are given in table 15.1. You probably know that during blood transfusion, any blood cannot be used. The blood of a donor has to be carefully matched with the blood of a recipient before any blood transfusion to avoid severe problems of clumping, that is destruction of RBCs. The donor's compatibility is also shown in the table 15.1. From the above mentioned table, it is evident that group O blood can be donated to persons with any other blood group, and hence O group individuals are called universal donors. Persons with AB group can accept blood from persons with AB as well as the other groups of blood. Therefore, such persons are called universal recipients. 15.1.3.2 RH grouping. Another antigen, the RH antigen, similar to one present in rhesus monkey, hence RH is also observed on the surface of RBCs of majority, nearly 80% of humans. Such individuals are called RH positive, and those in whom this antigen is absent are called RH negative. And RH negative person, if exposed to RH positive blood, will form specific antibodies against the RH antigens. Therefore, RH group should also be matched before transfusions. A special case of RH incompatibility mismatching has been observed between the RH negative blood of a pregnant mother with RH positive blood of the fetus. RH antigens of the fetus do not get exposed to the RH negative blood of the mother in the first pregnancy, as the two bloods are well separated by the placenta. However, during the delivery of the first child, there is a possibility of exposure of the maternal blood to small amounts of the RH positive blood from the fetus. In such cases, the mother starts preparing antibodies against RH antigen in her blood. In case of her subsequent pregnancies, the RH antibodies from the mother can leak into the blood of the fetus and destroy the fetal RBCs. This could be fatal to the fetus or could cause severe anemia and jaundice to the baby. This condition is called erythroblastosis fetalis. This can be avoided by administering anti-RH antibodies to the mother and immediately after the delivery of the first child. 15.1.4 Coagulation of blood. You know that when you cut your finger or hurt yourself, your wound does not continue to bleed for a long time. Usually the blood stops flowing after some time. Do you know why? Blood exists coagulation or clotting in response to an injury or trauma. This is a mechanism to prevent excessive loss of blood from the body. You would have observed a dark reddish brown scum formed at the site of a cut or an injury over a period of time. It is a clot or coagulum formed mainly of a network of threads called fibrins in which dead and damaged formed elements of blood are trapped. Fibrins are formed by the conversion of inactive fibrinogens in the plasma by the enzyme thrombin. Thrombins in turn are formed from other inactive substance present in the plasma called prothrombin. An enzyme complex thrombokinase is required for the above reaction. This complex is formed by a series of linked enzymic reactions cascade process involving a number of factors present in the plasma in an inactive state. An injury or a trauma stimulates the platelets in the blood to release certain factors which activate the mechanism of coagulation. Certain factors released by the tissues at the site of injury also can initiate coagulation. Calcium ions play a very important role in clotting. 15.2 Lymph tissue fluid. As the blood passes through the capillaries in tissue, some water along with many small water-soluble substances move out into the spaces between the cells of tissues, leaving the larger proteins and most of the formed elements in the blood vessels. This fluid released out is called the interstitial fluid or tissue fluid. It has the same mineral distribution as that in plasma. Exchange of nutrients, gases, etc. between the blood and the cells always occur through this fluid. An elaborate network of vessels called the lymphatic system collects this fluid and drains it back to the major veins. The fluid present in the lymphatic system is called the lymph. Lymph is a colorless fluid containing specialized lymphocytes which are responsible for the immune responses of the body. Lymph is also an important carrier for nutrients, hormones, etc. Fats are absorbed through lymph in the lacteals present in the intestinal villi. 15.3 Circulatory pathways. The circulatory patterns are of two types, open or closed. Open circulatory system is present in arthropods and mollusks in which blood pumped by the heart passes through large vessels into open spaces or body cavities called sinuses. Annelids and chordates have a closed circulatory system in which the blood pumped by the heart is always circulated through a closed network of blood vessels. This pattern is considered to be more advantageous as the flow of fluid can be more precisely regulated. All vertebrates possess a muscular chambered heart. Fishes have a two-chambered heart with an atrium and a ventricle. Amphibians and the reptiles except crocodiles have a three-chambered heart with two atria and a single ventricle, whereas crocodiles, birds and mammals possess a four-chambered heart with two atria and two ventricles. In fishes, the heart pumps out deoxygenated blood which is oxygenated by the gills and supplied to the body parts from where deoxygenated blood is returned to the heart, also known as single circulation. In amphibians and reptiles, the left atrium receives oxygenated blood from the gills, lungs, skin, and the right atrium gets the deoxygenated blood from the other parts of the body. However, they get mixed up in the single ventricle which pumps out mixed blood in complete double circulation. In birds and mammals, oxygenated and deoxygenated blood received by the left and right atria respectively passes on to the ventricles of the same sides. The ventricles pump it out without any mixing up, that is, two separate circulatory pathways are present in these organisms. Hence, these animals have double circulation. Let us study the human circulatory system. 15.3.1 Human circulatory system. Human circulatory system, also called the blood vascular system, consists of a muscular chambered heart, a network of closed branching blood vessels and blood, the fluid which is circulated. Heart, the mesodermally derived organ, is situated in the thoracic cavity in between the two lungs, slightly tilted to the left. It has the size of a clenched fist. It is protected by a double-walled membranous bag pericardium enclosing the pericardial fluid. Our heart has four chambers, two relatively small upper chambers called atria and two larger lower chambers called ventricles. A thin muscular wall called the interatrial septum separates the right and the left atria, whereas a thick walled, the interventricular septum separates the left and the right ventricles, figure 15.2. The right atrium and the ventricle of the same side are also separated by a thick fibrous tissue called the atrioventricular septum. However, each of these septa are provided with an opening through which the two chambers of the same side are connected. The opening between the right atrium and the right ventricle is guarded by a valve formed of three muscular flaps or cusp, the tricuspid valve, whereas a bicuspid or mitral valve guards the opening between the left atrium and the left ventricle. The opening of the right and the left ventricles into the pulmonary artery and the aorta respectively are provided with the semilunar valves. The valves in the heart allows the flow of blood only in one direction, that is from the atria to the ventricles and from the ventricles to the pulmonary artery or aorta. These valves prevent any backward flow. The entire heart is made of cardiac muscles. The walls of ventricles are much thicker than that of the atria. A specialized cardiac musculature called the nodal tissue is also distributed in the heart, figure 15.2. A patch of this tissue is present in the right upper corner of the right atrium called the sinoatrial node SAN. Another mass of this tissue is seen in the lower left corner of the right atrium close to the atrioventricular septum called the atrioventricular node AVN. A bundle of nodal fibers, atrioventricular bundle AV bundle, continues from the AVN which passes through the atrioventricular septa to emerge on the top of the interventricular septum and immediately divides into a right and a left bundle. These branches give rise to minute fibers throughout the ventricular musculature of the respective sides and are called Purkinje fibers. The nodal musculature has the ability to generate action potentials without any external stimuli, that is, it is auto-excitable. However, the number of action potentials that could be generated in a minute vary at different parts of the nodal system. The SAN can generate the maximum number of action potentials, that is 70 to 75 times per minute, and is responsible for initiating and maintaining the rhythmic contractile activity of the heart. Therefore, it is called the pacemaker. Our heart normally beats 70 to 75 times in a minute, average 72 beats per minute.